Welcome back to the second video about transcription. This video will differentiate bacterial and eukaryotic transcription. The first difference between the transcription processes of a eukaryote and a prokaryote comes with the steps that they follow. Prokaryotes follow the simple steps of initiation, elongation, and termination. While eukaryotes have a more complicated process, it has two general steps. After initiation, elongation, and termination, there is a production of the pre-mRNA, but this is not yet ready for translation, so it has to undergo RNA processing to make the final product, which is mRNA. Let us start the discussion with bacterial transcription. The first step of bacterial transcription is initiation, and in this process is a promoter. A promoter is made up of short DNA sequences or target sequences. These are the ones that are recognized and where the RNA polymerase will attach to. A promoter is located before the initiation site. So if this is the site where transcription should happen, a promoter would be seen upstream or before that site. An E. coli promoter has two components, the -10 and the -35 positions, named so because they are located at the -10 position and the -35 position after the +1 site. These are also known as the -10 box and the -35 box. The +1 site is made up of around 20 to 600 base pairs located upstream of the start coding region. The -10 box is also known as the Pribnow box, which is named after the scientist that has first discovered it. The -35 box is the one recognized and bound to by the sigma subunit of the RNA polymerase. The negative or the -35 box has a sequence of TT, GA, CA, while the -10 box has a sequence of TA, TA, AT. The most efficient promoters are named as strong promoters, and these promoters are 1,000 times more productive in initiations as the weakest promoters. The RNA polymerase in E. coli plays a special role in the initiation process. This is a large protein made up of different polypeptide units. The core enzyme is made up of two alphas, a beta, and a beta prime subunit. When the sigma subunit is attached to the core enzyme, we call this now as the holoenzyme. An E. coli has around 7,000 RNA polymerase enzymes, and 2,000 to 5,000 of these enzymes are actively participating in transcription at any one time. The second step in the bacterial transcription is elongation, where there is RNA synthesis or the creation of new RNA. Elongation starts when the sigma subunit dissociates itself from the core RNA polymerase. Once the core is free, it can now start synthesizing RNA from the 5' to the 3' direction. An RNA polymerase may synthesize up to 40 nucleotides per second. As the RNA polymerase synthesizes, it moves forward and continuously unwinding the upstream of the DNA. As it creates the transcript and moves forward, the DNA part that it leaves behind is also continuously rewound. The RNA polymerase, or the bacterial RNA polymerase, can cover from 30 to 40 base pairs. And inside the RNA polymerase is the transcription bubble, which can hold around 12 to 14 base pairs. In this transcription bubble, where the RNA and the DNA strand are together, can be held at around 8 RNA and DNA base pairs. The elongation process of the bacterial transcription is not continuous. It is described to be a discontinuous synthesis with rapid elongation and some pauses or some brief pauses. They are brief because they only last around 6 milliseconds each. There is also backtracking or the going back or the reversing of the RNA polymerase every now and then. An RNA polymerase has a polymerization rate of around several hundreds of nucleotides per minute. The last step in bacterial transcription is termination, which signifies the end of the transcription process. This starts when the bacterial RNA polymerase dissociates itself from the DNA. This dissociation would then liberate the newly formed RNA. We call this process as the termination of transcription. In bacteria, two strategies are used, the intrinsic termination and the rod-dependent termination. One common feature of these two strategies is that there are inverted repeats in the termination end. Inverted repeats are sequences that are followed by its reverse complement. In the intrinsic termination, these inverted repeats would cause the formation of intrastrand base pairing, and this base pairing would cause the formation of a hairpin loop. Now this hairpin loop, once formed, will start the termination sequence. We also call this as the intrinsic terminator. The rod-dependent termination, on the other hand, uses a protein called RAW, which attaches itself to the RNA transcript. This protein will move upwards towards the RNA polymerase. Now, since this raw protein is also a helicase, it can break the DNA and the transcript. So as it moves forward towards the DNA or the RNA polymerase, it would break down the DNA and the RNA transcript from each other, therefore releasing the transcript itself. And once the transcript is released, this would then signify that the termination process is complete. Let's now start the discussion with the eukaryotic transcription. Transcription in eukaryotes is more complicated when compared to bacteria. This is because eukaryotes have a more sophisticated mechanism in terms of gene expression. Specifically, there is more attention given to the preliminary process when the pre-mRNA is formed. The first step in eukaryotic transcription is initiation, and one of the complexities of eukaryotes is that the RNA polymerase cannot recognize the promoter of the DNA directly. It needs the help of several transcription factors. And the first of these transcription factors is the TF2D, or the transcription factor 2D. This is the one that makes the initial contact with the DNA. The TF2D is made up of the TATA binding protein, or TBP, and the TBP associated factors, or TAF. The TBP is described to have a saddle-like appearance, as it is able to wrap itself around partially on the TATA box of the DNA. Once the 2D has attached itself to the DNA, another factor will help out, which is 2B. It attaches itself to the 2D so that it can form a structure that can be recognized by the RNA polymerase. Once the 2D and the 2B are attached to the DNA, the RNA polymerase will recognize this structure, and it can now start binding with the help of another factor, which is 2F. This is the one responsible for binding the RNA polymerase. And this also prevents any wrong placement of the RNA polymerase in the other parts of the DNA. After 2F, we have two more factors, the 2E and the 2H. And once these factors have attached, we now have the helicase, which is in charge of breaking down the DNA into two parts. And with these two factors attached to the assembly, we now can say that this is the complete initiation complex. This initiation complex should be activated by the addition of phosphate. Now, once it has been activated, the RNA polymerase can leave the complex and start the transcription process. The difference between the RNA polymerase used in eukaryotes and in bacteria is that eukaryotes have three types of RNA polymerase, as bacteria only have one. The three types are the polymerase 1, Pol 2, and Pol 3. Now, although these three are related in their structure as they somehow look the same, they have very different functions. Researches are more concentrated on the RNA polymerase 2 because this is the one in charge for the gene coding of proteins, or as we say, this is the one responsible in the transcription process, although it also transcribes small nuclear RNA and microRNA. The other two polymerase, polymerase 1 and polymerase 3, can only transcribe non-coding RNA. Next are the promoters, and these are DNA sequences that are recognized by the RNA polymerase for the initiation step. For eukaryotes, we have the core promoter, and this is the site or the place in the DNA where the initiation complex, the one that was just described earlier, and the RNA polymerase would assemble. So in this DNA sequence, it would be in this site where the TF2D would bind to the Tata box. So this is the core promoter. There are different promoters for the three different types of RNA polymerase. So for the promoter of polymerase 2, this is considered as the most complicated, as it can be seen several kb upstream of the gene that should be transcribed. It also has the Tata box promoter, where the Tata binding protein would attach or would start the initiation process for the initiation complex. The Tata box is also the counterpart of the Pribno box in bacteria. The promoters for polymerase 1 and polymerase 3 are not as complicated as the promoter for polymerase 2. In addition to the promoters in eukaryotes is the alternative promoters. These type of promoters allow more than one active site. So these are the promoters that allow multiple transcription processes happening in a single DNA strand. The second step is elongation. Bacterial and eukaryotic elongation are generally the same. This is the synthesis or the creation of the new RNA transcripts. The RNA polymerase builds from the 5' to the 3' direction, so that means the new nucleotides should be attached to the 3' end of the DNA template. DNA also runs anti-parallel with each other. The difference now comes in the following information. For bacteria, it can only take minutes to copy a single gene, while in eukaryotes, a single gene may be copied in a few hours. Bacteria can copy hundreds of nucleotides per minute, while in eukaryotes, up to 2,000 nucleotides can be copied per minute. Also, in eukaryotes, we have multiple introns that allow multiple transcription processes happening in a single DNA. This can show the length of the DNA for each organism. There is a much considerable length in a eukaryotic DNA compared to a bacterial DNA. That's why even though it can copy thousands of nucleotides per minute, it still takes hours for a single gene to be copied. And the third step for eukaryotic transcription is termination, where a termination site is seen. And this termination site would be different for the different RNA polymerase that is used. So for example, for the RNA polymerase 1, we use a DNA binding protein. This DNA binding protein is seen after the genes that should be transcribed and is usually located 12 to 20 base pairs after the genes transcribed. For RNA polymerase 2, we add a poly-A tail. A poly-A tail is a series of adenosine nucleotides that are attached at the termination site. And unfortunately, not a lot is known for the RNA polymerase 3 termination. Now we are done with the different steps in the transcription process, but as we have mentioned earlier, for eukaryotes, there is another additional step. So we're done with initiation, elongation, and termination, but after these three processes, we have only made a pre-RNA. So we still have another process, which is known as the RNA processing, so that we can come up with the final mRNA that would be used in the translation process. The fourth and last step in the eukaryotic transcription is the RNA processing. This step is not seen in prokaryotes because the mRNA made after termination is ready for translation, whereas in eukaryotes, the mRNA made after termination still needs to be processed before translation. We call this mRNA as pre-mRNA or as precursor mRNA. There are three steps in the RNA processing. The first one is the addition of the cap and tail. The second one is the removal of the introns, and the third one is to join the RNA together. When we say cap and tail, we are referring to short amino acid sequences that are added before and after the RNA template. The 5' cap is made up of modified guanine, while the 3' tail is made up of a series of adenine nucleotides. Both the cap and the tail are added in the free ends of the RNA to serve as its protection, since enzymes are detrimental for the RNA template. The second step is the removal of introns. Introns are the part of the genes that are non-coding regions. That means they do not have the codes to create any proteins. They are still copied in the transcription process. That's why we have to remove them after termination. We retain the exon that has the codes for the creation of proteins. This is done by the use of an enzyme called spliceosome in the process named as RNA splicing. And lastly is just to join the RNA together so that we can have a complete mature mRNA. This mature mRNA is what we will be used for the next process, which is translation. And that ends this video about the bacterial and eukaryotic transcription. Next would be the regulation of transcription. Thank you very much for watching.